So good morning. And so today's session is all about the final dispatch and distribution, where the medicine actually gets into the goes to the clients for further processing. That's for drug substance, or it gets onto the shelf. So before that, I just wanted to share with you, I will put it in the format that is required. The Syngene format. I had prepared this for another lecture of mine. And just put it in the format and share it with Dr. Vasan so that he can upload it. So if you look at the case study related to Abbott infant formula recall, there have been many recalls recently. I don't know if you've heard of the another one, that is Johnson's baby powder recall, because it had asbestos in it, which is again not relevant and not to be put into medicine. Okay, so let's look at Abbott now. In February 22, Abbott Nutrition, very, very famous for products, nutritional products related to various brands, very well-known brands. Some of it are manufactured in India, like Ensure, Pediasure. So what happened is they announced a voluntary recall. This is in the US, not in India. They announced a voluntary recall of some of their infant formula brands, and they included Similac, Alimentum, and LAKSHMI. And Similac is a common infant formula brand. Because of its recall, it affected many babies and led to a nationwide formula shortage. So this is when the government agencies like the FDA, DCGI are very alert of such instances. Why? Because it can affect the population of the country. And their mission is to protect the population of the country. So in this case, there was a shortage of the formula, and it was manufactured in Sturgis, Michigan. And of course, it didn't include other metabolic deficiency nutrition formula. So there's certain powder infant formula that was manufactured in that area in Sturgis in Michigan. And because it was widely sold in US and also to some countries, probably the Middle East and Southeast Asia. So again, this I picked it up from the FDA website. So this is where FDA is alert, and they put in these guidance documents so that the citizens of the country and also the healthcare providers like the doctors are alert, and they ask them to recall the product. So again, what they say is look at the code. FDA recommends looking at the code. It's a multi-digit code. And again, they have said, and not to use if such a code is there, or such a code on the container is there, and the expiry date is so much. All those details have been indicated there. See, just because they put it on the website doesn't mean everybody goes through it. So they had to alert the various pharmacies across US. Let's talk about US and not other countries where it got exported. So the distribution system in the US is very evolved as compared to, say, our own country, India. It's evolving now, but not as much as it has evolved there. That means they can trace and track the medicine consumed by the consumer, which we cannot do. Okay. When I say that, like, for example, if a person with hypertension or diabetes goes in for a checkup with their doctor, because it's highly based on the entire medical system or healthcare system is based on the insurance, they all pay insurance. And since they all pay insurance, they are covered by the insurance for certain tests and certain doctors, etc., etc. I mean, I will not get into the schemes there. And they go visit their physicians, for unlike where, we go to different doctors who are close by or probably there in the next road or recommended by somebody. Over there, they have to go to only those assigned by that area insurance as per the policy. So they go over there and the doctor will prescribe certain medicine, and they just cannot go to any pharmacy like the way you and I go. They have to go to that particular pharmacy, and they have to give this prescription, or they will already get it through the online system if they have. And when they go there, the medicine is given in a bottle, tamper-proof bottle. I'll speak about that bottle. Tamper-proof bottle, and say they will give a refill of 30 days. And of course, they will give a huge, leaflet related to the medicine. That means all the side effects or adverse effects of the drug is given to them. It's a big, thick leaflet. I've seen it. One of my uncles were carrying it, about sometimes as much as 30 pages leaflet they give them, because they need to understand what are the different causes. And that's the patient education that they do. So that thick leaflet is given and they take it home. After third, and they also in that on the label very clearly says to whom it is, what is the age, what is their Medicare number, whatever you call that. All those details are given in that bottle label. And of course, the expiry date, everything is given. So again, it also says refill due on, say 2 months from now or 1 month from now. Again, they have to go back taking that bottle and hand it over and get a new one. So this is how the US drug dispensing happens to the patient. Sometimes, of course, blister packs, etc., etc. But what I want to say is that they are able to track that tomorrow if they want to recall this Abbott and they have recommended this formula to a child in our family. So we would have got it from that again area, and they would have tracked it over there and said that you need to recall this back. Ship it back. All those instructions would be given. So how did it start? I started with bacterial infection after ingesting the formula, where the 4 babies became sick. All 4 involved Protobacter sakazaki infection. It can cause severe life-threatening infection, sepsis, or meningitis. It's rare but high risk for newborns. It includes poor feeding, irritability, temperature changes, jaundice, breath, abnormal breath, grunting breaths, and abnormal body movements. It occurred in Minnesota, Ohio, Texas. These are the states. And again, here, this is actually from the FDA website that I've taken, that the instructions are very clearly given there. And Abbott sent a message out to the nationwide saying that they routinely conduct testing for that and other pathogens in the manufacturing facility. And they did find this infection, bacteria in the plant in a non-product contact area. But however, they didn't find any salmonella, but however, the investigation was going on. And of course, later on, they were providing updates. You can check it out on the net. So some of the FAQs asked by a new parent, like, talking about, I'm having hard time finding the formula, baby infant formula. So what is the FDA going to do? So FDA is working with Abbott Nutrition in assessing the impact of the recall. That means they would see what happens is Abbott will not manufacture only from one site. They'll be manufacturing from the other sites as well. So what they will do is they will stop manufacturing there and sort of, allow the other sites to start manufacturing it in a much more higher manner, quantity rather. And again, they advise not to use homemade formula. Again, this is the FDA, this is for America, this is not related to India. But this is just to give an idea how the recalls occur. So let's move on to a Tylenol case study. So in the 80s, Tylenol, even today, is the most successful over-the-counter product, and 19% of corporate profits come in from there. However, in the 80s, what happened? They received a threat saying that there were deaths, and that was because of the Tylenol extra strength capsule was laced with cyanide. And the minute they heard that J&J, that is Johnson & Johnson, recalled the Tylenol across America. across America. The minute the death was declared, it was due to cyanide that was laced in Tylenol that was done. So you can look in the website about Tylenol cyanide deaths and case study. More details are available for you. And what happened was they did extensive investigation. They did extensive investigation in the plants. They did an extensive investigation in the stores. They looked at all those days, whatever camera they had, doubtful if they were cameras, but they tried to find out who could have, done that. And they rounded up all the suspected people, employees, etc., etc. But till today, they have not found out who did this. And considering this case, they decided to add an extra layer of tamper-proof seals. This is when the tamper-proof seals came into the picture. Why am I talking about it? I'm talking about what all things happened much before what the kind of tamper-proof seals that we see now. Why are they doing that? So that it's not tampered with, unnecessarily like this, and it's causing harm to the patient. Some may argue saying that it's not the responsibility of J&J, but ultimately the product is J&J's, the packaging is J&J. So it is their responsibility to ensure that the correct medicine goes to and the strength goes to the patient. So it is their responsibility ultimately. In fact, all the government agencies like FDA, the police, everybody along with J&J sort of, came in together to verify what happened and all that, but they were clueless. Very, very sad instance. There was another instance of the odor. If you remember, we did discuss about bleachable, extractable very briefly. So in 2008, there were some, see what happens, issues do happen in plants. If it's not appropriately identified, it can lead to bigger problems. So this is a classic example of that. And they complained of, again, Tylenol, digestive problem, musty odor, and J&J and McNeil is also J&J's brand. And they resisted, FDA kept telling them to recall the product, but however, they resisted. But then they pushed for it, and then they realized that, yes, it needs to be recalled. So they recalled Tylenol for 3 cases, 2 for musty odor and taste. 7 recalls happened in 2010, again, musty odor. And what happened was FDA gave them a warning letter. So whenever FDA comes for inspection, if they see inconsistencies, they give us a 483. That means a non-compliance, saying that this is what you had to do, this is what you didn't do, as per the law. But this, if you notice, they had already alerted J&J to recall in 2008. They didn't do it. 2009, they've done it, but it may not be that effective because in 2010, it still was creating problems. That means the investigation was not appropriate. That's when FDA gave them a warning letter. That means it's like a very high stringent letter, like saying that, listen, you've not done what we said. This is impacting the patient. You need to do it. So that's the, you could say, thought process that happens for a warning letter. And in case, if it's an Indian company, definitely they will stop the shipment entering America. And they will also seize it if required. Okay. Why do they seize it? Why do they use the term seize? Please understand these are legal terminologies which a common man may not understand. When they hear the word seize, they're like, oh my God. Yes, it's a oh my God case. But why are they seizing it is something you and I need to understand. They're seizing it so that it does not get consumed by a patient and again lead to complications. Okay. Now, coming to 2008, the phantom recall revealed during congressional testimony. So what happens is when J&J started doing this recall, what the head of the organization did was, this is known as the phantom recall. What apparently the CEO did was, he asked the people in his team way below, that is the representatives or the sales peoples, he told them to go. I mean, when I say he, it was in a letter. to the managers, and they in turn told their teams. That's how it got cascaded. They told them to go and go in to a shop, I mean, to go into a mall, pick up the Tylenol. Tylenol is like Crocin. So Tylenol from the shelf, and say that, you're taking it for your family, etc., whatever it is, and bill it and get the bill and get it reimbursed. That means they were without informing FDA. You see, you're supposed to, there is a process in which a regulatory recall happens. It's not just because you and I decided product has to be recalled. CEO says, get it out from the, what do you say, pharmacy. That's not how it happens. You have to inform the FDA or the DCGI here. So what happened here was, in this case, they told the employees or their contract employees also, saying, go to the shop, pick up, bill it, give the bill to us, we will reimburse you. Okay. So this nationwide, they did it, and the government came to know that this is what they did. And this is where the congressional committee, that means it went to the parliament, you can say. And that's where they were questioned. They were sort of, inquired as to why they did it and how can they, do this kind of issue, or rather a clandestine operation. Again, if you notice, it's going on 2011. And what again FDA did was, they realized that this is not going on well. They issued a consent decree. Consent decree is above the warning level, wherein they said that if you would like, see, one thing you need to understand is Tylenol is something that is consumed by all the citizens. Like, say Crocin, it's Tylenol is like that, and it's consumed by everybody. And sudden stoppage of that medicine can lead to, again, shortage of that medicine. So again, they want of that particular plant. They want the J&J. They put in a consent decree. They said you have to do 5 years of third party oversight. That means the consultants were there with the FDA, visiting them quite off and on. And they reviewed the entire process. And they said it's like third party. That means an independent consultant team is coming to review that entire process of the plant. And they did a sort of rectification. CAPA was done. Corrective and preventive action was taken. And again, if you realize, there's so many millions and billions of dollars were wasted because that many million packages were recalled, and they received 2 warning letters and closure also. And of course, they were under criminal investigation because, as I said, they went secretly, and they withdrew all the Tylenol from the various shops, and their CEO, Bill Weldon, resigns. So this continued, and finally they identified the cause. It was 2,4,6-tribromoanisole, that is TBA. It's a bimethylation byproduct of 2,4,6-tribromophenol. It is TBP is used as a preservative and antifungal in many warehousing, shipping, and retail operations, and it can readily contaminate plastic and gelatin containers. So this is where they identified the cause, but it took a long time for them to sort of, you could say, identify the cause. So please understand, it is much more than, till now we had heard of manufacturing issues. We had heard of, so many other complications that happened during the manufacturing and all those things. But you also realize that it can be something else as well. So companies have SOP on product recall, and product recall is different from customer complaint. So initially, it starts with a complaint coming in from the citizens or from the patients, and that needs to be investigated in a methodical manner that is considered as a customer complaint, and we process it. But if in case such instances, what you saw, serious instances happen, also over there, they would have indicated in what cases they will do a recall. The company has to trigger a recall. Okay. So I move on to the next aspect that is related to distribution. Um, Distribution requirements. So good distribution practice, it indicates the minimum standards that the distributor must meet to ensure the quality and integrity of the medicines is maintained throughout the supply chain. That means the medicine may be manufactured anywhere in India or abroad, and by the time it reaches the designated area, it still should be having that particular quality and integrity of the medicine. And they have to verify, companies go and verify whether those distributors have the necessary infrastructure. That means the warehouse. If in case it's cold shipment, they verify if there is a good cold shipment over there. All these are sort of, verified before they are given the contract to handle it. And some of them also is to ensure that adequate controls are there to prevent mix-up, adulteration, contamination, cross-contamination, and spurious. So one of the things is spurious drugs, which is, like counterfeit drugs, as they say. That's one of the threats that the industry faces. So many companies have their own barcoding systems. They have their own, they have their own, what do you call, controls in place so that it's appropriately addressed. And
Me,
why do
ma'am.
we need to follow? Yeah.
Ma'am, you had already given an assignment for today on storage conditions for distribution.
Okay. So will you take it up?
No, ma'am, actually, sorry, we forget that in the middle of this mock interviews. Sorry,
Okay.
ma'am.
So how is the mock interview going on?
Yeah, we had completed our one round of HR and technical session, and we have one more on Saturday on
Oh.
HR round.
So when is the placements going to be?
15th,
That is next Wednesday.
next Wednesday.
Okay. Next Wednesday, you'll all be sort of gearing up. Yeah. No, it'll all work out fine. Don't worry.
Okay, ma'am. Thank you.
So no problem. But did I go through these slides?
No, no, no.
Okay. Thanks for telling me. Yeah. It is to protect the product, and the procurement, purchasing, storage, distribution, transportation, documentation, and record keeping are very, very important elements, though it may be a few, but it is so. It applies to everyone in the industry who is responsible for the storage and distribution, from the manufacturer to individual dispensing, and or giving it directly to the patient. So sometimes what happens in the hospitals also, they have a dispensing area, especially in clinical trials. There is a pharmacy or hospital pharmacy that is the one that will dispense the medicine to the patient. And it also applies to the sourcing, storage, and transportation of the API and other ingredients. That is both DS and DP. And again, it also involves wholesalers, suppliers, distributors, government organizations. In fact, some cases, especially for BPL, below poverty level, government organizations are also involved in it. Again, international procurement organizations, the donor agencies, certifying bodies, logistics people like the courier services, the traders, the transport firms, forwarding agents, that means the shipping, they who help in shipping as well. All of them are responsible for this. Other than you and me, who will be involved in the manufacturing aspect, this is the rest of the value chain, rest of the supply chain, who are responsible for it. Again, there are various regulatory bodies. Like, for example, if from India we have to ship to America, there is a requirement. If you have to ship to EU, there is a requirement, regulatory requirement. Then there is a requirement coming from British, that is UK, then Japan, etc. It goes on. This is where regulatory teams are responsible. So they have a similar approach. Though it is similar, there are some slight variations based on the geographic area they are in. Okay, and personal quality system, premises, documentation, traceability, complaint return, and transportation. quickly we'll go through the couple of them. When it comes to principles, again, as we already said, national legislation or the rules must be ensured that the process is taken care of. The distributor whom we give it to should be legitimate. They should have a registered business. They should have the necessary infrastructure. And only authorized import or export agency should be handling the import-export of pharma. And they can only carry out distribution in nationwide when it is legal to do so. And all of them will have delegated responsibilities and roles. And again, distributors, so what happens is when they are given a license to operate or they have the authorization, it'll be given only to a particular class of product. So in some cases, they are Schedule H restricted drugs, like psychiatric drugs. So those will have a special permission. They'll need special additional controls. So it is not given to all the pharmacies. And also some drug products like immunosuppressants, oncology products, also companies control it. They distribute it only in case of when a prescription is given to that particular company, and the company ships it directly to the patient. That's also there. Okay, this is some of the controls pharma companies have put in, so that spurious drugs do not get mingled with the authentic ones. And the subcontractor, that is the vendor who is handling this entire thing, should be authorized, and only registered or approved mail order pharmacies should be able to sell medicinal products online. So again, if you notice, there are a lot of online apps that are there to buy medicines, right? In those cases, again, it is authorized, they have to be authorized to do it. Initially, the government was not willing to do it. This was, I'm talking about 7-8 years ago. But over a period of time, it has become the norm now. But however, the government still has control on it. They do not sell products like Schedule H, psychotropic drugs and all that. And again, there should be a person who's responsible, who's ultimately responsible for the distribution. And they should be competent, experienced, trained, and qualified. Training should be there. Hygiene, health, and clothing should be appropriate. Again, appropriate roles and responsibilities and their interfaces should be well defined. Code of conduct and punishments to be in place, because we've seen wherein sometimes the authentic drugs are replaced by spurious drugs, okay, counterfeit drugs. And all the documents need to be periodically reviewed. Designated person to handle the quality system should be there, and authorized procurement and release. Of course, conduct audits and evaluate the risks routinely. So this is about the quality system. When it comes to the warehouse and the premises, again, good storage practices, then you need to have authorized access only and prevent unauthorized access. Dedicated area for hazardous, quarantine, and rejected products.
Excuse me, ma'am.
First, yeah.
I'm sorry to interrupt. You have completed this topic, ma'am, at last class.
Okay. So,
Yeah, she completed. Oh, ma'am completed. Ma'am said about the overall traceability, importation, broker, everything she covered.
okay. Some of the elements are similar to that of storage, because this has premises and warehouse. Storage also has premises and warehouse. Okay. So some of the elements are there in both instances. So let me quickly go through it. Anything specific that you can think of asking me, you can ask. Yeah, probably I may have gone through it briefly, asking you to work on the assignment. Okay, so when it comes to documentation, it should be very clear and appropriately documented. Traceability of products distributed to facilitate recall should be there. Okay, and involve all stakeholders in the supply chain. This is where I was telling you in the developed countries like US and Europe also, they have very well laid out traceability. Now it is slowly coming into India and it will take some time, of course. And any complaints, recalls have to be investigated. Procedures should be there. Physical segregation and storage of return stock from the saleable stock should be accounted. So everything should be in place. And of course, it has to be traced. The vehicles, again, have we discussed this by any chance, Prashanti?
Yes, ma'am. Just a brief you said, that's all.
Yeah. So a couple of slides more and then we can conclude it, not a problem. So any vehicles that are used must be, again, taken care of to ensure that there are no rodents, insects. That means pest control should be good. And wherever relevant, special storage conditions should be provided. And the product should be safe. And they do have GPS systems, tracking devices. And dedicated vehicles must be used for transportation. Again, third-party carriers, distributors, they should have, excuse me, they should have the necessary permits and licenses. So we have the active ingredient supplier, and again, it goes through manufacturing, distributor, the pharmacy, and to the patient, or from the manufacturer, it goes to repackager. And they in turn may go to distributor and then go into the pharmacy, or they themselves may send it directly to the pharmacy. So there are various permutations and combinations that happen. So advantages are complaints are reduced, expensive mistakes are handled appropriately, consistency is there, wastage is reduced, and again, sustainability and effectiveness is maintained, and the right product gets delivered to the patient. And again, the design of the vehicles and everything should be kept in an appropriate manner. A written cleaning program should be available. And again, clean from vermin and rodents and all that stuff. So repackaging and relabeling, there are people who do repackaging and relabeling. And these are the folks who take the medicines, probably they may buy tablets directly, and they may package it as a separate what do you call activity, or chances are that if I'm a company, I might outsource it to you to manufacture it and make it as a label as per my logo and sell it and send it to me, and I will in turn sell it to the distributor. So there are various permutation and combinations that happen, but during the inspections, it is so well traced. It is so well sort of, you could say, tracked to verify from where did it come and who's the manufacturer and all that. So next time you consume a tablet or a medicine, just have a look at the label. In the label, because it gives you more idea about the label. I request you to do it in the coming days itself, so that you're much more clear about what are the various aspects that go into the label. If you actually see at the end of the day, the product label gets approved by the drug authorities. And when you say product label, ultimately it is the product also that gets approved by the regulatory authorities. And any counterfeit drugs are taken, it has to be segregated from other products and recorded, and of course informed to the regulatory authorities. And let's get into the FDA aspect. So when it comes to what do you say, importation, it should be restricted by suitable laws. It should be stored under suitable conditions. And then the contract activities are, again, if you outsource it to another party, you still have to be in control. That is the thumb rule. You may outsource it to others, but you still have to be in control. And again, wholesale distributors should ensure all means are available to minimize the risk of falsifies a medicinal product entering into the legal supply chain. And in EU also, by a wholesale distributor must be covered by a marketing authorization grant by EU or the member state. Again, for broker, these are people who are involved in activities in relation to the sale or purchase of medicinal product. that do not include physical handling and consists of negotiating independently on behalf of other legal or natural persons. So there are brokers as well. Brokering agencies, they are called as. Yeah, I come to the end of the session. Any questions? So, no questions, not a problem. You know where I'm available. I'm available online in case you have any doubts, any clarifications, anything. And take care of your forthcoming interview sessions, your exams, and definitely more importantly, on top of the list is your health. Okay, and I wish you all the very best. And it was enjoyable interacting with you. And we did interact in person as well. Okay, I know I can see a lot of anxious faces here. But I can see the same anxious faces will be smiling couple of months later when you come for your graduation ceremony. I can see the change in the body language that happens, the way you people will be in the coming months. So this is just to give a heads up that things will work out. I'm sure you will get into good profiles. Okay, don't worry. Think clearly, answer carefully, listen very well. Very importantly, study your basics. Well, we have taught you the industry practices. So one of the questions I get asked routinely is, we are biochemists, molecular biologists, microbiologists, but what is this industry requirements versus us? So what do you think it is? These are basically guiding principles for you to execute your scientific knowledge in the industry. That's all I can put it. Right? You may join different departments. You have the knowledge, you are subject matter experts in your various techniques, technology that you have studied, scientific principles you have studied. Please study your basics well. Go through the notes, whatever we have given, make your salient points. Okay. Very importantly, in the interview, try to behave like a solution provider. That's when you will be able to correlate many concepts together. Like, for example, they may ask you, okay, this batch failed, what will you do? And you will say, we did that, we can do this, we can do that, then we have to take corrective action, we have to address it, record it, etc., etc. So once you think of in that line automatically, that means you understand the principles of the industry as well. Okay. Not taking too much of your time. All the very best and have a good day
2
and
minutes,
have a
ma'am.
good test and interviews.
2 minutes, ma'am.
Yeah.
Okay, so on behalf of this SMF batch 1 students, I'm taking this moment to express my immense gratitude to our beloved Lakshmi ma'am. I would like to say heartfelt thanks to you, ma'am, for making us capable to get fit into this big industry. For sure, by next week, we are going to face our interview, and we are thrilled about that actually. And you did a really great job in training us, and you did a hard work for making us to get into to face this interview, and also for trained us to perform in the interview also. Along with the lecture sessions, you instilled us with the confidence and made us more and more ambitious. Your classes, your experiences regarding the industry you shared with us will always be our stepping stone. We could proudly say that we are not in the same level when we came here before 4 months. Thank you so much. You played a big role in it. Once
Thank you.
again, thank you from the bottom of heart, ma'am.
Thank you. All the very best.
Thank you, ma'am.
Take care.
Thank you,
Bye.
ma'am.
Thank you, ma'am.
Thank you.
Bye.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank
Thank
you,
you,
ma'am.
ma'am. Thank you, ma'am.
Hello, Vishal sir. Hello.
Yeah, good morning. Good morning, team. Is anyone there?
Good morning, sir.
Good morning, sir.
Yeah. Am I audible?
Yes, sir.
Yeah. So today's topic, I understand, is on two topics which have been given. What are the why are change rooms needed, and what is the positive pressure and air changes required for? Am I right? These are the two topics which are given for today. Hello.
Yes, sir.
Yes, sir.
Yeah. So we start. So let us take the first topic: why change rooms are needed. Okay. Now, I think last time my colleague Sheshadri has introduced you regarding the clean rooms and also the requirement of change rooms, airlocks. So now we dwell a little deeper into it. It's a very small topic, but still it is interesting in terms of why it is required. So the change room, as the name suggests, is for changing. What for? See, whenever a person enters into a clean area, he has to change it to a gowning facilities. Okay. The display ಇದೆ ಯಾವ್ದು? This is positive pressure. Yeah. Yeah. Are you able to see the screen?
No. No, sir.
You're not able to see the screen. Hmm. I think that. Are you able to see the screen?
Yes, yes.
Yes, sir.
Okay. Now. So there, I am joined by my colleague Seshadri. We both will take the course together. He will share a part of it, and he will share. He will discuss some of his experiences. So let me start with the fact that why change rooms are needed. As the name suggests, change rooms are a part of the clean room facilities. What? Why we require a change room? Uh, because clean room requires proper gowning, because this ought to prevent any contaminations, microbial and particle contaminations. Human bodies, when they go directly into the clean areas, they develop the particulate contamination and the microbial contamination. To prevent that entering into the very clean areas which are into the which are going for the manufacturing areas, we require a change room. Change room, as the name suggests, it literally means an area where there is where the personnel who is entering into the room changes into some clean gowns or sterile dresses, so that the particulate and the microbial contaminations are reduced. So the bioburden or the particulate load in the clean room areas are reduced by wearing a proper gowning. That's what is the uh, area where the dresses are changed, or the sanitization, cleaning, wearing a head cap, wearing the gloves, wearing the shoes, mittens. All those things are done so that the particulate counts and hence the bioburden counts due to the normal human activities is reduced, and hence the areas which are going for the productions are very clean. So that is the basic requirement of a change room. Now that is the now then there is another thing called an airlock. Next. Airlocks are commonly used in pharmaceutical industry to maintain cleanliness and prevent contamination of the manufacturing environment. When I say like this, these are a buffer zone between the outside environment and the manufacturing facility, so that the it's a pressurized area, airlock, so that any outside contamination will not be able to pass into the manufacturing environment, which is a very clean facility. Now that is what is called an airlock. Now usually the airlocks are provided at the entrance to a manufacturing facility along with the change rooms, so that the outside environment is not contaminating or a direct access to the production facility. Now you might ask me a question: what is the difference between a change room and an airlock? Correct, that's the question, because both are doing the same function. In one, the personnel changes to a gown which is more sterile, more clean, and other thing. The airlock on the other side also does the same function. It prevents the outside environment coming directly in contact with the manufacturing facility. As the name suggests, both are having the same function, but it do serves different purposes. Changing rooms are places where people entering or leaving a clean room can change into or out of clean room apparels. Means a person who is entering into a manufacturing facility has to wear a gown, head scarf, nose protection, goggles, and all those things. The place where it is held, stored, and where it is worn is called a change room. Similar to that change room, there is an airlock, but airlock doesn't have this function, but it prevents, it is a buffer zone. It is not, it is a place between the outside and the manufacturing area where there is a lot of pressure, air pressure, to prevent any contamination entering directly into the manufacturing thing. So this is the difference between change room and airlock, though they serve the more or less the same function. Till now, I hope I have made myself clear to you. If there is any questions, please ask me on these two things, because this is the main topic for today.
Have they visited this facility?
You asked during. Seshadri is also there, so together we are conducting this class. Seshadri, you want to talk to them?
During this 2 months or 3 months, your experience in the pharmaceutical industry, have you visited a change room or a airlocks?
Yes, sir.
Yes, sir.
Sir.
So,
Yes.
what is your observation on a change room, and what is your thoughts on a change room and an airlock? Let me understand so that we can clarify it to you, or you are very clear about it.
What is that you have observed when you have entered a change room there?
Sir, we already did a gowning in the initial months of training. So
Okay.
we went to injectable areas in Sinjin, only in the corridor. So
Okay.
we had gone through a gowning process. So in change room will be provided with gowns, mask, gloves, head cap, everything. And there is a crossover bench there, and there is a protocol through which we should we should do the gowning. And after the gowning, we have to cross that bench and do the sanitation, and we'll enter to the corridor. At that time, we have permission only to enter into the corridor only.
Correct. So now you understand the necessity of the gowning is only to protect the environment, the production area, to protect
Yes.
that area from the dust generated by the occupants.
Yes.
Because the humans are the most contaminating resource in the any manufacturing sector. You understand this? Yeah.
Yes, sir.
Yes.
Yeah. So have you also seen an airlock when you entered Syngene
Yes, sir.
injectable area?
Yes, sir.
Yes, sir.
So what was there in an airlock from your point of view?
It's a high pressure flow of air.
Correct. So there was not that airlock is not meant to change your apparels or gowning procedures, but it is an airlock which is just provided to prevent the outside air coming into it, either by over pressurization or by providing a sink. So that we will talk about what are the different types of airlocks where these air coming slides. Okay, so that's for now. This we talked about the changing rooms thing, which we have already gone through the process, and now the in the change room or an airlock, there will be one entry door and one exit door. Have you also seen that once you enter, you cannot unless and until both one door closes, the other door is not opened. Have
Yes,
you
sir.
observed
Yes,
that?
sir. Yes, sir.
Yes, yes, sir.
Now that is called as interlocking. Why? Because a person who is coming in, if he opens the other door, it becomes a via passage again that to for the contaminants and other things to enter. Now that's why there is an interlock. Once the door is opened, the other door till it is closed, the other door will not open. During that process, the person changes the apparels and the pressure inside the change room in terms of the airflow, number of air changes will be higher, so that all the dust contaminants will be taken away by the return air. So that's the purpose of a change room and an airlock, because it is a air. It's an area where you are flushing a lot of dust contaminants through the human beings or by means of whatever products or the equipments which you are taking into it, by pressurizing that room by a high number of air changes into the room and taking it out to the return air risers. So that is the thing. So door interlocks will be provided, and because the rooms have to be pressurized to a certain extent compared to the outside. So that is one thing. You would have seen that also, right? That there will be an interlock and people will not be able to get out if at all the other door is open and other things. And also, then the sanitization procedures happen in the change rooms. What is the sanitization procedure? Then sterilization of articles. You could have seen that solvents, the hand, what do you call that?
Yeah, IPA.
IPAs which are kept there, and that you have to scrub your hands, and then you have to enter into the other areas. So I think there are two slides which show about that. Seshadri, if you can explain them, what is the process? This one, sterile clean room learning process. Yeah, this one.
This is the basic
So gowning procedure,
gowning
typical
procedure,
gowning procedure.
gowning procedure, which is showing you to have a boot covers. Basically, if you don't change in ready area, there will be only a boot cover will be provided. When you go to the core area, then entire will be stripping down and will be wearing a sterile full gowning. So it starts with our head cover, shoe cover. These two are the basic thing, so that we will be covering the dust which will be carried through our shoes into the facility.
Correct. Okay.
First in the
Okay. Sorry. How it goes? How it? Inga. Sorry for this. Okay. Mm. Why is not shared? Someone is said to share that. No? I think you can come here and control it. Sit here. Sit here. I will be adding some points. Don't. I will add some points.
Is the screen visible now?
Yes, sir.
Yes, sir.
Yes, sir.
So this basically gives you a glimpse of the steps how to cover our entire sterile gowning procedure. So here, first thing is covering the head and head and toes, and second is cleaning our hands and cleaning with the IPA and other sources, and using a gloves for the entire head cover, and this is for the sterile suit. Then over and above that sterile suit, we would have an rubber shoes provided within the facility, and this is the second set of the gloves and the goggles. That is, this entire thing is for a sterile environment for before entering a sterile environment. So these are the other removable type sterile garments, whatever we have should be discussing now. These are the outer gloves, goggles, boot covers, coveralls, hoods, mask, inner gloves, ear net, and other shoe covers. Thank you.
So we talked about the gowning procedures, and
Yes, sir.
so I think they have got an experience of changing the gowns when they have entered into the system.
Yes, sir.
So now we should talk about airlocks. Now, can you talk about this airlock, different types of airlocks and everything?
Bubbles in the next.
You'll explain what about this airlock. Bubble and explain. Mr. Moore, that's all. Finish this and take a break, and then we'll do. Are you having that presentation there?
Yes, sir. Yeah, I already done it.
Okay. So as we have discussed, I think Seshadri have explained.
We have already explained this
during,
cascading during our HVAC. Just we have to give them a brief on it.
Okay. So there are different types of airlocks to keep the different pressure. Sometimes the airlocks are used as a negative point because the production, for example, in a tablet area, the production area will be having a lot of dust generated compared to the outside. So the airlock which is in between has to be in the negative pressure. That is called a sink. Now somewhere where we have to keep the airlock higher than the production facility, that is called a cascade or a bubble airlock. Now, somewhere we have to have a airlock which will keep the pressures right from the corridor with a plus to an airlock having plus plus, and the clean room to be the very cleanest area, which is triple plus. So when you say plus, it is representing the pressure, 5 Pascal or 10 Pascal, which is the delta P or the pressure differential between the each rooms. Now, there are three different types of airlocks which we have highlighted here. One is the cascade airlock, wherein it maintains a midpoint between the high pressure area, which is the cleanest area in a, say, for injectable grade A or B. The airlock is the in between pressure compared to the cleanest pressure, which is at the highest pressure into the manufacturing of a vial filling or an ampule filling. Compared to a storage area. This picture which is depicting here, you see this is the injectable area. If you see the manufacturing area, the airlock will be maintained at one pressure lesser than the clean room, because the air should always go out from the clean room towards the airlock, not from the airlock to the clean room. Otherwise, the contamination will again enter into the clean room. So this is the airlock, and this airlock should be having a positive pressure compared to the outside corridor, which is very dirty. So therefore, this is called a cascade. This is the cascade from plus to double plus to triple plus. This is called a cascade airlock. This is mostly in the injectable facilities, which we get into this kind of a cascade airlock. Now, there are certain areas wherein we have to maintain the pressure on both sides, corridor as well as the clean room, on the same pressure, because we cannot overpressure the system. Then we have called something called a bubble airlock. The airlock which is inside, in between those two, will be a higher pressure. So the air from airlock will go out to the corridor and to the clean room equally, so that there is a positive pressure between the corridor and clean room, and hence there is no contamination happening between the outside environment to the manufacturing areas. So this is the bubble airlock.
Neither of the two areas.
Which one?
Contamination.
Contamination is neither going into the clean room nor into the corridor, but the airlock is very critical here. Okay, next slide, please. Then there is something called as a sink airlock. Now, in a sink, it can do it either way, because when keeping the airlock double pressure compared to corridor, or the airlock will act as a sink, because from the corridor also the contamination will come, and from the clean room also the contamination will be coming into the airlock, and from there it is going back to the air handling unit. So this is called a sink because it receives the contaminated air either from the corridor and from the clean room and takes it out of the thing. So thereby this airlock acts as a negative pressure compared to corridor and clean room. This is these are the three types of airlocks. What are the next?
Only one.
So the next. How does an airlock work? Just for the summary part of it is the airlock. Next. Airlock creates a controlled enclosed space with two interlock doors. They make a controlled transitional space between external environment and a clean room. When you say an external environment, in a clean room facility, it will be the corridors, either the dirty corridor or the material movement corridor, like that, with the primary goal of preventing contamination, maintaining cleanliness, and preserving the specified environmental conditions within the clean room. So next slide, Seshadri, because this slide is done. Can you please take it out? So then, how does an air? As we have explained, there are three types of airlock. One is the cascade, or other is the bubble and the sink. So how does it work? You always saw that there is a plus, 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 and plus, triple plus. So that is a pressure differential. The clean room typically operates at a higher pressure than the surrounding area. Normally, it operates on a higher pressure. But in the case of tablet manufacturing, wherein there is a lot of dust which is created due to the compaction or due to the powder coating or coating activities, there will be a lot of dust generated. So that will be kept negative compared to the outside. Only in that case it will be negative. Even in onco drugs, oncology products productions, because the substance is highly contagious, so we cannot be able to exhaust it or put it in a positive pressure. So hence that room will always be in the negative compared to the outside airlock or the environment. So these are the two examples where it will be changing. Otherwise, more or less, it is always the clean room areas are positive compared to the outside. When personnel need to enter or exit the clean room, they first enter the airlock. The outer door is closed behind them, sealing of the outside environment. This is an explanation of how the door interlock happens. Now, based on the pressure which we have put in terms of the internal and the external airlocks, the number of air changes and hence the pressure inside the airlocks compared to the corridor as well as the production area will be defined. Number of air changes, pressure differential, exfiltration, infiltration. I think these are the terms which you would have heard during the earlier lecture conducted by my colleague Seshadri in the HVAC part. So those things will be affected by an airlock design. Okay. So then when we use a personal procedures within that, that is called a change room. That is what we are always airlock when there is a personal protection like gowning and other thing, that is called as a airlock is called as a change room. That's the only thing. Okay. There are certain areas wherein in the airlock or in the onco facilities we have a mist shower or an air shower. Why? Because apart from the gowning activity, even the micro particles which are present on the gowns which we have put also should be removed. Hence, the person has to pass through an air shower or a mist shower. So that is also a part of the airlock or the change room facility in a clean area. So these are the summary of what is the change room, what is the airlock, how it works. And I think it's a very small topic, so hence we couldn't elaborate it on a very bigger scale. We had only these two, change room and airlock, so we were able to put in some slides and explain to you. Please let me know if you have any queries on whatever we have talked. We have talked from our experience only. If based on your queries, if you have any questions, we can be able to at least look at how to answer it.
They can explain where they can use this bubble sink.
Seshadri has a question to you. So can you tell me, in the facilities what you have seen in Sinjin or wherever you have gone through, whether it was a cascade or a bubble or a sink airlock? Anyone?
Bubble, sir.
Okay. How did you know it is a bubble? A good question, good answer, but how did you know it is a bubble?
Now you explained as it is we entered for the injectables. So you informed about the pressure that it will be same, but at the hairlock the pressure will be more.
So in a injectable, if you look at it, that is a cascade type of an airlock. Why I say that is the injectable manufacturing. Suppose it's a vial or an ampule or any other filling area, that should be at a very very high pressure. Not even a single contaminant, even from the airlock, should get into it. That means that the production is in a triple plus area, plus, very, very high pressure. So the air which is pressurized has to enter into somewhere, it has to exit. So it will go into the airlock. So the airlock is at a slightly higher pressure compared to the outside, but lesser pressure compared to the manufacturing. So that's why it is called a cascade. Means plus plus, again if you can go back to that slide wherein we can explain, or it is triple plus, airlock is double plus and outside is single plus. That is the question, that is the answer for that cascade. Oh no, what she was trying to explain is because it's a bubble, but bubble will not be in case of manufacturing of an injectable steroid.
And one more thing, have you observed how many airlocks were there when you have visited the facility?
Two, sir. One is personal hairlock and one more is material hairlock.
Those two are, you are right. One exclusively, there will be two entries, one for men movement and second will be for metal movement. But in men movement also, have you, any one of you counted how many change rooms are there?
Two, sir.
So usually when you are entering into any core manufacturing area, sterile manufacturing area, so there will be 5 change rooms, 3 on the entry path and 2 on the exit path. Have you noticed this?
In a sterile area,
Yes.
when we are saying injectable.
Yeah, it's sterile manufacturing.
Okay, so 3 on the entry and 2 on the exit. So
Yes.
have you noticed it? That's one of the first question, because it's a very important thing that he will explain to you why that 3 plus 2 will be there. See,
So
the process is like this. When you enter into a sterile, the first is the initial street dress will be removed, correct? Then, after that, they will collect the sterile gowns from the
LAF unit.
LAF unit, means under the laminar airflow unit, it will be kept. So that is the care that they take. He will collect it. He will remove the street dress. He will collect the, in the second change room. He will collect the gowning, sterile gowning, and then he will go to the third.
he will wear the entire sterile gowning in the second.
And
He
then
will have the first set of his shoe cover and hand glossing done in the second change room.
third.
In the third change room, he will go, when he enters inside, he will cleanse his hands, the gloves. Above that, he will be wearing the goggles, head caps, and the second gloves before entering into the core manufacturing area.
That is the entry procedure for the sterile 3 change rooms. So first is the street dress, undressing, and then he goes into the collects the sterile from the lab, which will be very, very protected. He will wear that, and the third is wearing the peripherals like the gown, the caps, and additional gowning also. Sometimes it is required. Otherwise, it is some. These are the specific procedures of that particular company, which will be placed in a thing, which will be written in a part of SOP, and then only he will enter into the manufacturing. While returning back
On the first, that is the fourth change room, you will be once again degowning, and in the fifth change room, you will collect his street cloths or the factory garments. You will wear that factory garments and you will come out.
So these procedures are very critical during the sterile entry and exit. Whereas in a OSD, it might be a single change room. When in a sterile or an injectable, these procedures have to go through it. And in fact, if you go to an onco production, onco products and a very hazardous product, he has to go through one more change room wherein he has to go for a wet shower. First is the undoing of the outside dress, shower. Then he goes into the lab, collects the document, and then he enters into this room. So these are the another one more change rooms will be added. So this is, for example, what we were explaining to you. Any other questions on these things?
So I have a doubt.
Yeah.
So for the sterile, the clean room will be having the high pressure, more high
Yes.
pressure than the other one. So we'll be using the cascade airlock. So, but in the slide when you mentioned for the bubble airlock, no, so you have given us example as injectables. May I know why?
No, bubble airlock usually they will use in OST because the corridor and the production areas, if you are having 2 or 3 types of tablets being manufactured in the same facility adjacent to each other, there should not be any cross contamination. There should not be any cross contamination between the areas.
But she's asking about this, why we have written this, because the critical part will be the final filling and manufacturing. what we are explained to you verbally when we told you is that the critical final manufacturing is the filling, ampule or a vial filling.
This is a concept.
This is a concept.
This is the concept where we are showing you only one airlock. Now, as we have explained you, there is a series of change rooms and airlock coming in between the areas. So these airlocks will be provided specifically to avoid the cross contamination. Whereas these change rooms are basically it is required for any entry and exit procedure.
Her question is for that. Okay.
Correct. This is a airlock. Airlock between any process area or between two areas.
So what we explained to you is for the final ampule or a vial or any filling area.
Okay, sir.
Is it clear?
Yes, sir. Thank you.
Sir, what will be the entry procedure for high-potent API production units?
That is what sir was explaining right now. High potent area is similar when compared to your oncology drugs or any other thing. High potent area, one thing, the cloning procedures will be same, only except for the on the way back they will be going for an air shower so that whatever the particles are on the gown will be de-dusted there, and then they will come out, they will degown. After that they will go for a shower, mist shower or an directly shower itself. After shower they will come out and they will wear their factory garments.
One more changes, as you rightly asked, in when you are talking about the high potent thing, they go with the suits, means the completely protected suits wherein none of the body parts are exposed. It's like it has got its own breathing apparatus, means the compressed air breathing apparatus, because you are not going to be exposed to the high potent drug when you are inside. Otherwise, the process, as what Seshadri has explained, is there, except that it will be a suit, means like a space suit like that. He has to wear a suit with a compressed air or a breathable air apparatus, which will be provided, or it will be there. You can hook it up so that you can breathe inside that suit. There should not be any exposure to the potent product or the API. That's the only change which is happening. Otherwise, the process of getting in, getting out is more critical, because the product, the cloth has to be discarded.
Thank you, sir.
Any other points?
It's a very airlock and change room is a small topic. So, but it's an interesting topic which is very critical to the clean room facility. So that's where we come from. Only when you practice, when you go into it, and each of these companies will have its own, change room procedures which are written down, and that's how you have to follow that, and that's what they will be validating it for.
Whatever we have explained is the concept, and each facility will have its own SOP, and based on the number of airlocks, number of change rooms, change procedure, the pressure gradient will be differentiating, and based on the pressure gradient, there will be a use of all these things, the cascading bubble and even the sinks.
Sometimes the type of airlocks will also be based on the type of air handling units which you use, or the type of air conditioning systems which we have to use. The philosophy governs the overall type of airlocks design, and whether it is cascade or an air sink or an air bubble. There are different approaches to designing an airlock from the HVAC perspective.
Any other thing?
So we take questions. If it is okay, then we take a 5 minutes break, and then we start the next topic.
Okay, sir.
Is it okay, or you have some other questions? You can always ask me or Seshadri, that we can be able to explain or come back to you with that. So we assemble at 11:05.
Okay, sir.
Okay. Next topic in there.
Yeah. Can we start or
Yes, sir.
still
I'll talk to them.
We can start, sir.
Yes, sir.
yeah. Now the next topic is about positive pressurization and negative pressurization. Okay. I'll be taking this subject because our Mr. Sundar is not keeping well today. If any additional points, he is here only, he will be guiding us. Okay. Shall I go ahead?
Yes, sir. Yes, sir.
Yeah. Till now, whatever we have discussed regarding the airlocks and change rooms, do you have any question on that?
No,
No?
sir. No, sir.
Okay, fine. Can anyone answer what is positive pressure and what is negative pressure before we go into the next slides? Sorry. What is the pressure, at least, which we have all studied in 6th standard, I think?
Yes, sir.
Ah,
The force exerted by air per unit area.
correct. Okay. And we have also studied that the pressure is exerted equally in all direction.
Yes, sir.
Okay. Now, what is meant by positive pressure and what is negative pressure?
Sir,
Sir,
the
higher air pressures,
Yeah, go ahead.
than the
higher air pressures are positive, sir. Whereas
Pardon?
higher air pressures are positive,
Okay.
uh, and lower air pressures are negative.
Okay. Coming to our subject, when we are maintaining a particular room by in a manner positive pressure or a negative pressure, how is that we are going to achieve that?
sir, the area high the area is in higher pressure when compared to other area. It is positive pressure.
No, I'm not asking to compare with any other room. Just with one room, when we are telling that when this room is positively pressured or this room is negatively pressured, what does it
Yes.
exactly mean?
Means particle won't enter to the positive pressure area.
Correct. All those things are right. We are not talking on the particle part, neither with respect to the adjacent areas. Just with respect to that particular room, when we are telling this particular room is positively pressured or this particular room is negatively pressured, what is the simplest mechanism of telling that this room is positively pressured? Okay, let me answer. See, whenever we are talking with respect to these rooms, we have been discussed on the HVAC part the other day. There we are based on the requirement of the room or the classification, we'll be pumping the supply air, right? The simplest example is that when we are supplying more air and taking less air, that means exhausting less air, the room will be positively pressurized. For example, if I am pumping 100 CFM into a particular room and exhausting out 80 CFM, what will happen to the remaining 20 CFM? Priya, can you answer this? Anyone? Omkar? When I am pumping in 100 CFM into a room, it's a very simple calculation, right? And I am exhausting and I am taking out 80 CFM. What is the remaining?
20, sir.
20 CFM. Yes, that's the right answer. Who is this? Prasantini. Very good. Okay. This 20 CFM of air is existing within the room. Every minute there will be 20 CFM extra. Then what will happen to this room? That will be pressurized, right? What will happen to this extra air volume? This extra air volume will try to leak from this room towards the other gaps, wherever the gaps are there, between the doors, between the windows, wherever we have gaps, that particular this air, because of the over pressurization, will try to go out. I'm clear till now? That means to say this room is positively pressurized. So now negatively pressurized. What should we do? Reverse of that. If I'm pumping in 100 CFM into the room, I have to exhaust 120 CFM. Then what will happen? This room is always short of 20 CFM. At that particular time, what will happen? This 20 CFM will come into the room through the gaps, wherever through the windows or doors, wherever it is, will get the air into the room. Thereby, this particular room is negatively pressurized. Till now, I am clear. Pramila, do you have any doubt? Pramila DB?
No, sir.
No. Okay. So, in your particularly when we talk about designing of our basic house itself, which area will be keeping at positive pressurization, which area will be keeping at negative pressurization? Can anyone answer? Omkar, Prathamesh, Shashank KS.
The kitchen will be in positive pressure and bedroom or all will be negative pressure.
Which area will be negative?
Other than the kitchen, sir, like hall or kitchen, hall or bedroom.
Okay. Basically, the answer is we don't require any positive pressurization in any of our house. Any of our rooms need not be positively pressurized. But negatively pressurized, definitely the kitchen and the toilet should be negatively pressurized. You understand? Correct, right?
Yes, sir.
Yes. Are you in agreement with me or not?
Yes, sir.
Yeah. Now, the design requirement, negative or positive pressure rooms are often necessary to prevent the contamination and maintain the climate controlled environment in various applications. While these rooms are somewhat similar, there are certain differences between the design and requirement, which I have just now explained you, how to maintain a room positively pressurized and how to maintain a room negatively pressurized. Okay. Here we will have a overview of both of these types of environment and their applications. Okay. So negatively pressurized, which is the area you nowadays for last 3-4 years you are very much aware that negatively pressurized rooms we were using in our hospitals. Aditi, can you answer this? Subhadeep, Omkar, Neeraja, all you people have underwent the COVID times, right?
Yes, sir. Yes, sir.
Yeah, then you people must be aware of this. The COVID centers were kept at a negatively pressurized. Why is it so? Hmm? Because of that contamination should not come out and infect the other people outside. Correct? This has to be an
Yes, sir.
interactive session. Hmm?
Yes,
Am
sir.
I right or not?
Yes, sir.
Yes, sir.
Okay.
Sir.
Negative pressurized rooms have their pressures that is lower than the external air pressure. Negative pressure is achieved by use of an exhaust system that frequently includes high efficiency particular air, that is HEPA filter. Why is it we are using HEPA filters here? So that this any contamination when we are exhausting the air out should not go out and infect the other people. That is the reason we are using these HEPA filters, so that we will be filtering up to 0.3 micron. All the contaminants shall be filtered, and even if it is required we can go use the scrubbers also. These rooms can trap a potential dangerous particles prevent cross-contamination into external air. Okay. These are basically used for isolation areas. These isolation area, the best example is the COVID centers. The person or the doctors or the healthcare workers who are entering the COVID centers, you might have seen how they were entering. You have to answer, my dears. They were entering with the full gowns, right? All the healthcare workers were in full gowning to protect themselves against the contamination which is spreading through the air. Correct?
Yes, sir.
Yes, sir.
Okay. Now, on the other hand, when we have a positive room, what example you people can give for a positive room? The slide is visible, right?
Yes, sir.
On the other hand, the positive rooms will be used for a where we don't have that immunity. For example, when a person is undergoing the treatment of for cancer, the bone marrow transplantation, BMTU. In such wards, it will be pressurized positively because the outside contamination should not enter, because that particular patient who is inside the room will be immunocompromised. So whatever the diseases for such kind of immunocompromised areas, we'll be using this particular positively pressurized rooms. Will be pumping in more air and will be exhausting out less air, thereby will be maintaining the positive pressure inside, and hence the outside contaminations will not enter inside the particular room. Am I clear?
Yes, sir.
When it comes to your same negative pressure areas, we'll be using where we are using this whatever the drugs which are formulated before releasing it to market, they'll be using on the particular animals for the testing. Are you aware of such things?
Preclinical, sir.
Exactly, preclinical tests. So such preclinical tests, when they are doing, that will also be done in an isolated rooms, where which will be kept negatively pressurized or positively pressurized based on the type of examination they are doing or the experiment they are doing. Clear? Any questions till now on the positive pressurization and negative pressurization? Okay, now coming to the pharmaceutical facility with respect to this positive and negative pressurization. A clean room is a facility may consist of multiple rooms with different requirements for the contamination control. All rooms in the clean room facility should be maintained at static pressures sufficiently higher than the atmospheric pressure to prevent the infiltration by wind and other effects. But till this point, Sundar sir explained you in the earlier lecture itself. This entire facility we are pressurizing just to avoid any contamination or infiltration coming through the air or other spaces. The differential pressure should be maintained between the rooms sufficiently to ensure the airflow towards the progressively from cleanest space to the least clean space during the normal operation, and a period of temporary upset in the air balance as the door connecting two rooms is suddenly opened. That means to say, whenever we are opening the door, there will be a pressure nullifying effect. What will happen? The pressure suddenly drops, and as and when the door is closed and the pressure builds up, This is taken as the recovery time. What is the time required to get the pressure back into the set standard? So this is called as recovery time. Whenever a door is opened between the two rooms, the differential pressure will be zero. For example, if I am having a differential pressure of even 20 Pascal between the rooms, just by opening the door for 2 or 3 seconds, the pressure differential will come down. Then once the door, all the doors in clean rooms, if you see, it will be with a door closure, and the door opening will also be towards the higher pressure only. Why is it so? Omkar? Anybody? Because due to the pressure, if the door is opening on the opposite side, due to the air pressure, there will be always a difference of delta, and the air pressure will not allow the door to close fully. If the door is opening towards the positive pressure side, irrespective of the door will be closed because of the room pressure itself. Understood? I want to answer from your side.
Yes, sir.
Yes, sir.
Okay. All systems shall be designed to attain the desired pressure levels within the room relative to adjacent area. In general, the space differential pressure between the adjacent areas will be maintained by a fixed air differential, achieving the air balance of supply and return air. As I have already explained you, the room pressure will be basically based on the supply, what is that I am supplying into the room and what I am exhausting out of the room. The space pressure differential will be designed to cascade from the cleanest classified area to non-critical spaces. And as you are all aware, there is a standard called ISO 14644 Part 4. The pressure differential creates a barrier between cleaner zone towards the less cleaner zone. Higher pressure differential between adjacent zones shall be controlled and balanced and taken care to avoid turbulence. As per ISO 14644 Part 4, it states what is the minimum difference, that is Pascal pressure difference between the rooms. They initially they say the pressure can you see this value here? My cursor?
Yes.
Hello?
Yes, sir.
Yes,
Yeah.
sir.
So there is an example given here. There is room A and room B. The minimum differential pressure what the authorities say is requirement of 6 Pascal. Okay. For validation purpose, it is taken as plus or minus 2 Pascal on the designed value. That means to say, if I have designed room A at 25 Pascal, room B at 35 Pascal. So plus or minus 20, the acceptable value of room A is minus 2 Pascal or 2 plus 2 Pascal. Same in the case of room B, it is either minus 2 Pascal or plus 2 Pascal. From 25 Pascal, minus 2 Pascal value is 23 Pascal and plus 2 Pascal value is 27 Pascal. Even though these two rooms, one is at higher value and the second is at lesser value, the difference between these two, if you see, 33 minus 27 is the 6 Pascal. This 6 Pascal difference has to be maintained at any given point of time between these two rooms. Whereas if you see, we are talking about the same class and same HO, we will be keeping 10 Pascal difference between the rooms. Like room A and room B are both are at same class and same HO. What do you mean by class here?
Great, sir.
Exactly. It is the grade, whether we are talking about grade A, grade B, grade C, grade D areas. And what do you mean by same AHO?
Hair handling unit. So
Exactly. AHO is air handling units. Here, same AHO means these two areas. For example, we are talking about the first scenario. These two areas, room A and room B, are of same classification or grade, whether it is grade C, grade D, grade D. Then the difference, and both are catered by same AHO, then the allowable pressure difference should be 10 Pascal. You understood? If the both the rooms are catered by same HO, second scenario, but are different classification, that means to say this is grade D, room A is at grade D, and room B is at grade C, then the required pressure differential should be 15 pascals. Third scenario is that different class, different HO. Room A is scattered by different HO, room B is scattered by different HO. In the same thing, this is of grade D and this is of grade C. In that scenario also, we require 15 Pascal. When we are entering from a unclassified to classified area, definitely the requirement is 15 Pascal. That means to say, when room A or the corridor is unclassified and room B is a classified area, then at that particular scenario, instead of if this value is 25, then here the room pressure should be 40 because the delta required here is 15 Pascal. Okay? Any doubt till now? If you are not understood, I will explain once again. Hope this is clear to you. Same class, same HU, same class, different HU, different class, same HU.
Yes, sir.
Okay. Now, we'll be seeing a basic one simple example where if you see this red color hatch is scattered by one particular HO, this green color hatching is scattered by one HO, and this blue hatching is scattered by third HO. Is it clear till now?
yes,
Yes,
sir.
sir.
Yeah. Now this red color corridor, this is a corridor where this is scattered by a corridor HO, and the pressure here what we are maintaining is at 15 Pascal. As we have explained the earlier scenario, we have a vial filling and sealing area here for which the person will come in, he will just dig on here. We'll just talk about the pressures now. Let us not talk. I'll just brief you on the anyhow. The person will come here. He will dig on. He will cross over this bench. He will take the necessary precaution here for going inside. When he enters into the second change room, he will collect the clean garments from this particular cabinet. He will cross over. Then he will gown himself here. After that, he will sanitize himself here once again. Then when he is entering into the third airlock, he will get those goggles, head cover, then the second gloves, sanitize, and then he will enter into the vial filling area. On the way back, when he is coming here, he will discard all his gownings here, come to the airlock 5, pick up his factory garment. This cabinet is openable on both the side. Pick up his factory garments and he will wear that one. He will discard. You can see one dustbin here. We will discard all his gloves and other things here, and he will come out to the corridor. This is the gowning principle, entry and exit procedure. Whereas now coming to the pressurization part, this corridor is at 15 Pascal. Next, we are maintaining at 30. What is? Can you see this value?
Yes,
30
sir.
Pascal. So
30 Pascal. What is the delta here? Delta difference between this and this.
15.
15.
Exactly. Why 15? Because there is a change. Even though this is at grade D, For example, I'll tell you the classification also. Corridor will be at the grade D. This will be at grade D. Then this second airlock will be at grade C. Then this will be at grade B. While grade filling area will be also at grade B. Then once again, while coming here, this will be grade B, grade C, and the corridor D. Because there is a change in grade, and also the AHU, there is a different AHU. The minimum differential pressure to be maintained is 15 Pascal. That is how we have arrived at 30 Pascal here. Okay. Now the scenario here is same AHU between these two rooms, same AHU but different classification. Correct? From D to C. Okay. From D to C, we are maintaining 15 Pascal difference here. Then from C to B, there is one more 15 Pascal difference. Here, both these things are at the same grade. Even the while filling is at grade B, and HU airlock 3 is also at grade B only. Then why we are maintaining 15 Pascal difference here?
Different age.
Exactly. Very good. Who was that?
So it's me.
Who?
Sir.
Huh?
It's me, Joshua.
Johnson. Okay. Okay. So because we are these two areas are catered by a different HU. The delta is 15. Once again, on the way back, we are having a delta as 15 here because of the great difference. If you see here, the delta here, it is 30 Pascal. Why we are giving a 30 Pascal? Why this room cannot be at the 15 Pascal delta? That is why this value cannot be at 30 Pascal. Can anyone answer? On the entry path, we have 3 change rooms. On the exit path, we have 2 change rooms, right? Because our pressure gradient will be dependent on the way when we are coming inside, the more number of change rooms, that will dictate the pressure. Clear? This ends with the pressure gradient, positive pressure and negative pressures. Now, if you have any query on any of these slides which I have shown, you can ask me. I'll be able to help you out. No queries? Pramila DB. You understood it fully?
Yes, sir.
Vishnu Priya.
Yes, sir.
You can have your time. You can go through this document. I hope you have received all these documents. You can ask
Yes,
us
sir.
if you have any doubts tomorrow also. You are not received, is it?
No, sir, we didn't receive it.
Okay. Then Dr. Vasant may be sharing it to you.
Okay, sir.
Hmm?
Okay.
Well, hold on for a second. Just I wanted to brief you on the tomorrow's timing. Let me check with Mr. Sundar.
Okay, sir.
Tomorrow we'll start at 10 o'clock. Okay.
Okay, sir.
Thank you so much.
Thank you, sir.
Thank you, sir.
Thank you,
Thank
sir.
you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you.